8 Kelvin World Present. Here I will show you wildlife animals. Animals are multicellular, eukaryotic organisms in the biological kingdom Animalia. With few exceptions, animals consume organic material, breathe oxygen, are able to move, can reproduce sexually, and go through an ontogenetic stage in which their body consists of a hollow sphere of cells, the blastula, during embryonic development. Over 1.5 million living animal species have been described, of which around 1 million are insects, but it has been estimated there are over 7 million animal species in total. Animals range in length from 8.5 micrometers to 33.6 meters. They have complex interactions with each other and their environments, forming intricate food webs. The scientific study of animals is known as zoology. Most living animal species are in Bilateria, a clade whose members have a bilaterally symmetric body plan. The Bilateria include the protostomes, containing animals such as nematodes, arthropods, flatworms, annelids, and mollusks, and the deuterostomes, containing the echinoderms and the chordates, the latter including the vertebrates. Life forms interpreted as early animals were present in the Ediacaran biota of the late Precambrian. Many modern animal phyla became clearly established in the fossil record as marine species during the Cambrian explosion, which began around 539 million years ago. 6,331 groups of genes common to all living animals have been identified. These may have arisen from a single common ancestor that lived 650 million years ago. Historically, Aristotle divided animals into those with blood and those without. Carl Linnaeus created the first hierarchical biological classification for animals in 1758 with his Systema Naturae, which Jean-Baptiste Lamarck expanded into 14 phyla by 1809. In 1874, Ernst Haeckel divided the animal kingdom into the multicellular metazoa and the protozoa, single-celled organisms no longer considered animals. In modern times, the biological classification of animals relies on advanced techniques, such as molecular phylogenetics, which are effective at demonstrating the evolutionary relationships between taxa. Humans make use of many animal species, such as for food, for materials, as pets, and as working animals including for transport. Dogs have been used in hunting, as have birds of prey, while many terrestrial and aquatic animals were hunted for sports. Non-human animals have appeared in art from the earliest times and are featured in mythology and religion. All animals are composed of cells surrounded by a characteristic extracellular matrix composed of collagen and elastic glycoproteins. During development, the animal extracellular matrix forms a relatively flexible framework upon which cells can move about and be reorganized, making the formation of complex structures possible. This may be calcified, forming structures such as shells, bones, and spicules. In contrast, the cells of other multicellular organisms are held in place by cell walls and so develop by progressive growth. Animal cells uniquely possess the cell junctions called tight junctions, gap junctions, and desmosomes. With few exceptions, in particular the sponges and placozoans, animal bodies are differentiated into tissues. These include muscles, which enable locomotion, and nerve tissues, which transmit signals and coordinate the body. Typically, there is also an internal digestive chamber with either one opening or two openings. Animals are categorized into ecological groups depending on how they obtain or consume organic material, including carnivores, herbivores, omnivores, detritivores, and parasites. Interactions between animals form complex food webs. In carnivorous or omnivorous species, predation is a consumer-resource interaction where a predator feeds on another organism. Selective pressures imposed on one another lead to an evolutionary arms race between predator and prey, resulting in various anti-predator adaptations. Almost all multicellular predators are animals. Some consumers use multiple methods. For example, in parasitoid wasps, the larvae feed on the host's living tissues, killing them in the process, but the adults primarily consume nectar from flowers. Other animals may have very specific feeding behaviors, such as hawksbill sea turtles primarily eating sponges, hydrothermal vent mussels, and shrimps. Most animals rely on the biomass and energy produced by plants through photosynthesis. Herbivores eat plant material directly, while carnivores and other animals on higher trophic levels typically acquire it indirectly by eating other animals. Animals oxidize carbohydrates, lipids, proteins, and other biomolecules, which allows the animal to grow and to sustain biological processes such as locomotion. Animals living close to hydrothermal vents and cold seeps on the dark sea floor consume organic matter of archaea and bacteria produced in these locations through chemosynthesis. Animals originally evolved in the sea. Lineages of arthropods colonized land around the same time as land plants, probably between 510 and 471 million years ago during the late Cambrian or early Ordovician. Vertebrates such as the lobe-finned fish Tiktaalik started to move onto land in the late Devonian, about 375 million years ago. Animals occupy virtually all of Earth's habitats and microhabitats, including saltwater, hydrothermal vents, freshwater, hot springs, swamps, forests, pastures, deserts, air, and the interiors of animals, plants, fungi, and rocks. Animals are, however, not particularly heat tolerant. Very few of them can survive at constant temperatures above 50 degrees Celsius. Only very few species of animals inhabit the most extreme cold deserts of continental Antarctica. The following table lists estimated numbers of described extant species for the animal groups with the largest numbers of species, along with their principal habitats and free-living or parasitic ways of life. Species estimates shown here are based on numbers described scientifically. Much larger estimates have been calculated based on various means of prediction, and these can vary wildly. For instance, around 25,000, 27,000 species of nematodes have been described, while published estimates of the total number of nematode species include 10,000, 20,000, 500,000, 10 million, and 100 million. Using patterns within the taxonomic hierarchy, the total number of animal species, including those not yet described, was calculated to be about 7.77 million in 2011. Animals are found as long ago as the Ediacaran biota, towards the end of the Precambrian and possibly somewhat earlier. It had long been doubted whether these life forms included animals, but the discovery of the animal lipid cholesterol in fossils of Dickinsonia establishes their nature. Animals are thought to have originated under low oxygen conditions, suggesting that they were capable of living entirely by anaerobic respiration. But as they became specialized for aerobic metabolism, they became fully dependent on oxygen in their environments. Many animal phyla first appear in the fossil record during the Cambrian explosion, starting about 539 million years ago in beds such as the Burgess Shale. Extant phyla in these rocks include mollusks, brachiopods, onychophorans, tardigrades, arthropods, echinoderms, and hemichordates, along with numerous now extinct forms such as the predatory Anomalocaris. The apparent suddenness of the event may, however, be an artifact of the fossil record, rather than showing that all these animals appeared simultaneously. That view is supported by the discovery of Auroralamina attenboroughi, the earliest known Ediacaran crown group cnidarian from Charnwood Forest, England. It is thought to be one of the earliest predators, catching small prey with its nematocyst, as modern cnidarians do. Some paleontologists have suggested that animals appeared much earlier than the Cambrian explosion, possibly as early as 1 billion years ago. Early fossils that might represent animals appear, for example, in the 665 million-year-old rocks of the Trezona Formation of South Australia. These fossils are interpreted as most probably being early sponges. Trace fossils such as tracks and burrows found in the Tonian period may indicate the presence of triploblastic worm-like animals, roughly as large and complex as earthworms. However, similar tracks are produced today by the giant single-celled Protus gromia spherica, so the Tonian trace fossils may not indicate early animal evolution. Around the same time, the layered mats of microorganisms called stromatolites decreased in diversity, perhaps due to grazing by newly evolved animals. Objects such as sediment-filled tubes that resemble trace fossils of the burrows of worm-like animals have been found in 1.2 gyre rocks in North America, in 1.5 gyre rocks in Australia and North America, and in 1.7 gyre rocks in Australia. Their interpretation as having an animal origin is disputed, as they might be water escape or other structures. Animals are monophyletic, meaning they are derived from a common ancestor. Animals are sister to the Chonaflagellata, with which they form the Chonozoa. The most basal animals, the Porifera, Ctenophora, Cnidaria, and Placozoa, have body plans that lack bilateral symmetry. Their relationships are still disputed. The sister group to all other animals could be the Porifera, or the Ctenophora, both of which lack Hox genes, important in body plan development. These genes are found in the Placozoa and the higher animals, the Bilateria. 6,331 groups of genes common to all living animals have been identified. These may have arisen from a single common ancestor that lived 650 million years ago in the Precambrian. 25 of these are novel core gene groups found only in animals. Of those, 8 are for essential components of the WNT and TGF-beta signaling pathways, which may have enabled animals to become multicellular by providing a pattern for the body's system of axes, and another 7 are for transcription factors, including homeodomain proteins involved in the control of development. Several animal phyla lack bilateral symmetry. Among these, the sponges probably diverged first, representing the oldest animal phylum. Sponges lack the complex organization found in most other animal phyla. Their cells are differentiated, but in most cases not organized into distinct tissues. They typically feed by drawing in water through pores. The Ctenophora and Cnidaria are radially symmetric and have digestive chambers with a single opening, which serves as both mouth and anus. They are sometimes placed together in the group Coelenterata because of common traits, not because of close relationships. Animals in both phyla have distinct tissues, but these are not organized into organs. They are diploblastic, having only 2 main germ layers, ectoderm and endoderm. The tiny placozoans are similar, but they do not have a permanent digestive chamber. The remaining animals, the great majority, comprising some 29 phyla and over 1 million species, form a clade, the Bilateria, which have a bilaterally symmetric body plan. The Bilateria are triploblastic, with 3 well-developed germ layers, and their tissues form distinct organs. The digestive chamber has 2 openings, a mouth and an anus, and there is an internal body cavity, a coelom or pseudocolum. These animals have a head end and a tail end, a back surface and a belly surface, and a left and a right side. Having a front end means that this part of the body encounters stimuli such as food, favoring cephalization, the development of a head with sense organs and a mouth. Many bilaterians have a combination of circular muscles that constrict the body, making it longer, and an opposing set of longitudinal muscles that shorten the body. These enable soft-bodied animals with a hydrostatic skeleton to move by peristalsis. They also have a gut that extends through the basically cylindrical body from mouth to anus. Many bilaterian phyla have primary larvae which swim with cilia and have an apical organ containing sensory cells. However, over evolutionary time, descendant species have evolved which have lost one or more of each of these characteristics. For example, adult echinoderms are radially symmetric, while some parasitic worms have extremely simplified body structures. Genetic studies have considerably changed zoologists' understanding of the relationships within the bilateria. Most appear to belong to two major lineages, the protostomes and the deuterostomes. The basalmost bilaterians are the Xenocoelomorpha.